Good morning. This is Harsha Choudhary, pursuing my graduation from Dr. Hari Singh Gaur Central University, currently in BSc 5th Sem. Under the guidance of a professor, Dr. Dipali Jat ma'am, I am going to explain adaptive immunity, which is my topic. So, let's start with what actually is adaptive immunity. It is a type of immunity that develops when a person's immune system responds to a foreign substance or microorganism, such as after an infection or vaccination. Adaptive immunity involves specialized immune cells and antibodies that attack and destroy foreign invaders and are able to prevent disease in the future by remembering what those substances look like and mounting a new immune response. So this immune response or this adaptive immunity, it may last for a few weeks or month or maybe for a person's entire life. As we can see, the entire slide is divided into certain aspects of adaptive immunity. First and foremost, we'll speak about the characteristics of the same. So adaptive immunity displays the following characteristics. First and foremost is the antigenic specificity. So this property or characteristic of the immune system permits it to distinguish the differences among the antigens. Next, the immune system is capable of generating diversity in its recognition molecule, thus allowing it to recognize billions and millions of unique structures of the antigen. Once the immune system has recognized and responded to an antigen, it exhibits immunologic memory, which is the third characteristic. That is, the second encounter with the same antigen induces the immune reactivity. Last but not the least, the ability of the immune system to distinguish self from non-self molecules, which is quite essential. Next, we come to the group of cells which is responsible for adaptive immunity. An effective immune response involves two major groups of cell. First is the lymphocyte and second is the antigen presenting cell. The two major population of lymphocytes are B lymphocyte and T lymphocytes. The B lymphocytes mature within the bone marrow and each express a unique antigen binding receptor, which is the antibody molecule. So when a B cell that has not previously encountered any antigen encounters the antigen, it matches a membrane bound activity and the binding of the antigen to the antibody causes the cells to divide rapidly, thus forming its progeny, which is the memory B cell and the plasma cell. The memory B cells express the same membrane bound antibody as the parent B cell. The plasma cells have no such membrane bound antibody, but they secrete enormous amounts of antibody, which play a major role in humoral immunity. T lymphocytes also arise in the bone marrow, but they mature in the thymus gland. Now the T cell expresses a unique antigen binding molecule called the T cell receptor on its membrane. These membranes can recognize antigen that is bound to cell membrane protein, that is the MHC molecules. When a naive T cell encounters antigen combined with MHC molecule, the T cell proliferates and differentiates into memory T cells and various effector T cells. Thus, the two types or subpopulation of T cell are the T helper and T cytotoxic cells. T cell bearing CD4 and recognizing only antigen bound to class 2 MHC molecules are the T helper cells, and the T cell bearing CD8 recognizing only antigen bound to class 1 MHC molecule are the T cytotoxic cells. Now if we come to the types of adaptive immunity, there are majorly two types. First is the humoral immunity and second is cell mediated immunity. Humoral immunity is that aspect of immunity which is mediated by macromolecules. It is thus an antibody mediated response that occurs when antigens are detected in the body. This entire mechanism is primarily driven by B lymphocytes. Now, unlike humoral immunity, cell-mediated immunity does not depend on antibodies for its immune function. They are primarily driven by mature T cells, macrophages, and the release of cytokines in response to an antigen. Now, a question arises that how actually do these responses work? What is the mechanism. In cell-mediated response, after the T helper cell recognizes and interacts with an antigen MHC class II molecule complex, the cell is activated. It becomes an effector cell that secretes various growth factors collectively known as cytokines. These cytokines play an important role in activating B cells, TC cells, macrophages, and various other cells that participate in the immune response. Under the influence of T helper derived cytokines, a TC cell that recognizes an antigen MHC class I molecule complex proliferates and differentiates into a cytotoxic T lymphocyte, that is CTL cells. The CTL cell does not secrete many cytokines and instead exhibits cell killing or cytotoxic activity. Simultaneously, in humoral response, the released cytokine will induce B cells to divide rapidly, making thousands of identical clones of the B cell. As we have seen earlier, that these daughter cells either become plasma cells or memory cells. The memory B cells remain inactive, and later when these cells encounter the same antigen, they divide and form plasma cells. On the other hand, the plasma cells produce a large number of antibodies which are released free into the circulatory system. Now these antibodies will encounter antigen and bind with them and thus kill them. And in cell-mediated response, the activated CTLs recognize and kill the infected cells. Thus, this is how the entire mechanism of adaptive immunity works. This was all about adaptive immunity. Thank you for being a patient audience and giving your valuable time.